Greetings. Hello, and happy 55th Earth Day to participants at the Washington Native Plant Society Native Plant Appreciation Month 4th webinar of our series of 4. It is not the end of our events for Native Plant Appreciation Month, however, and we'll talk a little bit more about the BioBlitz at the end. But I'd like to welcome you right now to the 4th webinar for our series in our theme, Exploring Biodiversity Exercises in Community Science. We're hoping you can explore more about this by visiting the website, wnps.org. And we certainly would like you to, if you are a member, please accept our appreciation for your membership. Our mission is to promote the appreciation and conservation of Washington's native plants and their habitats through study, education, and advocacy, unquote. If you're already a member, we thank you for joining us. The membership supports plant conservation and education and connects us native plant enthusiasts across the state. Again, to learn more, please visit the website and consider becoming a member or making a donation. Your support is appreciated, and you will learn about webinars, classes, field trips, plant sales if you check into your local chapter site. We'll talk a little bit more about the BioBlitz, but check into the map of the Exploring Biodiversity BioBlitz, the umbrella for the Washington Native Plant Society, and there are lots of projects going on. You can check out your local area. Here in the Wenatchee Valley, we're going to be doing a little BioBlitz for beginners on Saturday. So hope to see some of you there. I'm also mentioning, I can't really focus on it, but as a membership benefit, you can get a print copy of Douglasia, which this spring issue has some wonderful articles, including Aaron Wells' article about five-needle pines. So just a little membership blurb there too. It's a great issue. I know it's not in focus here now because it's not my face, but there it is. My name is Cece Asplund. I'm a member of the Wenatchee Valley chapter of the WNPS, and I do want to acknowledge that I am speaking from the lands of the Wenatchee Pisquosa people, some of the original and continuing stewards of this beautiful area. Today I was privileged to be able to get up close and personal to our blooming serviceberry and our balsam root blossoms, and perhaps one day I'll learn how to pronounce them in their indigenous names. A couple of housekeeping items for tonight. Our webinar with Dr. Wells, we're going to be using the chat for comments. You should be able to see what you share in the chat function with others. And we will do questions at the end, so you can put a question in Q&A, and hopefully we will be able to get to it at the end of the presentation. So let me take another privilege here. I am very privileged to be able to introduce Aaron Wells, PhD. He is a vegetation ecologist and a project manager with the environmental planning and permitting team at AECOM Technical Services. His 20 years of experience includes designing and implementing natural resources inventory and monitoring studies, preparing natural resource impact analyses, and classifying and mapping vegetation and ecosystems across the Northern Rocky Mountains, the Pacific Northwest, and Alaska. Well, there sounds like lots of other stories that could be told from that bio. Last but not least, he is also a member of the Washington Native Plant Society, our WNPS, and in the Okanogan Chapter. So celebrating Earth Day today, and also the opening of the North Cross State Highway this week. It's now snow-free to get to the Okanogan Chapter from the west side. I'm very excited to be introducing Aaron Wells.
Well, thank you, Cece. I'm going to share my screen.
Great.
Okay, should all see PowerPoint. And can everybody hear me all right? Okay. Well, I'm gonna just jump into it then. Well, happy Earth Day, everyone. It's my pleasure to be here tonight and present on my citizen science monitoring project for 5 Needle Pines. I want to just start with some acknowledgements. Wanted to thank the Washington Native Plant Society for the grant funding that supported this pilot study. I'd like to thank the volunteer citizen scientists who assisted me on this project from Winthrop, Washington. They're there on the right. We had 6 volunteers. Sarah Goking, Forest Inventory and Analysis National Program Manager with the U.S. Forest Service, kindly reviewed my methodology and the report. Lonnie Chang, Agency Citizen Scientist Coordinator for U.S. Forest Service, also reviewed the report and helped assisted with the study design. And Carol Spurrier, an ecologist with the U.S. Forest Service, was also kind enough to review the methods and assist me with reaching out to the Forest Service in Winthrop. And I'd like to thank the Mettel Ranger District of the U.S. Forest Service for their service in managing our public lands. Let's see. I just skipped one. Okay. So my initial interest in 5 Needle Pines, what was it that drew me to 5 Needle Pines? Well, in 2000 to 2004, 2005, I was the project manager of a vegetation and soil survey of the Wind River Range in Wyoming, where I was introduced to whitebark pine, limber pine. Following that, I moved to Alaska for 14 years, where there are no 5 Needle Pines. My interest in 5 Needle Pines was renewed in 2021 when I moved to North Central Washington, and I started the Whitebark Pine Project. The website's there on my personal website. And I feel like I'm maybe missing a slide here. Hold on one second, everybody. Oops. Jumping ahead. There should be a YouTube video here, but it's not showing up. So I'm just going to jump out of the presentation for a second and go back in and see if I can see it again. There it is. So I just want to show you this video. It's a video I filmed when I was in Winthrop in 2023, and it's about my project.
My name is Aaron Wells, and I'm a vegetation ecologist. In 2021, I moved from Anchorage, Alaska to Washington's Mettel Valley. Since my move, I've been documenting occurrences of 5 Needle Pines in the North Cascade Range. Five needle pines are conifer trees in the genus Pinus that have 5 needles per bundle. There are 9 species of 5 needle pine that occur across the western United States and Canada, of which 2 species occur in the North Cascades. Western white pine, Pinus monticola, and whitebark pine, Pinus albicaulis. Six of the 5 needle pine species, including whitebark pine, are found in high elevation forests. Whitebark pine is a keystone species in the subalpine ecosystems where it occurs, meaning other species in an ecosystem will largely depend on it to such a degree that if it were removed, the ecosystem would change drastically. Whitebark pine has a mutualistic relationship with Clark's nutcracker, a bird that depends on seeds of whitebark pine as a major food source, and which plays a vital role in dispersing whitebark pine seeds across the landscape. A single Clark's nutcracker disperses tens of thousands of seeds per year and moves them up to 20 miles from the parent tree, where they store them in small caches. The seeds that the nutcrackers don't eat sprout into new trees. 5 needle pines are currently at risk of extirpation, resulting from a combination of climate change, pests and pathogens, and wildfire. White pine woster rust, a pathogen of white pines introduced to North America from Eastern Asia has been particularly devastating. For instance, in a 2018 article in the scientific journal Forests, the U.S. National Park Service reported that the portion of live whitebark pine infected with blister rust increased in North Cascades National Park from 32% in 2004 to 51% in 2018. And they concluded that the primary driver of whitebark pine decline in the park is white pine blister rust. Not all is lost for five-needle pines. Through concerted and coordinated efforts by state and federal agencies like the U.S. Forest Service, National Park Service, conservation groups like Whitebark Pine Ecosystem Foundation, the Nutcracker Ecosystem Project, and you, we can help create a more positive future for five-needle pines. Visit www.elfinwoodecology.org to learn how you can make a difference.
So that kind of outlines some of the reasoning and my interest in this project. So some other background here is just my interest in citizen science. My first introduction to it was using iNaturalist to document plant occurrences in Alaska. This led to the Flora of Chugach State Park project that I coordinated through my involvement with the Alaska Native Plant Society. I've been on the board of directors for 5 years now with the Alaska Native Plant Society. And the project objective was to prepare a plant species list for the 500,000 acre Chugach State Park that's near Anchorage. It's the 4th largest state park in the United States. And we used a combination of citizen science observations through iNaturalist and herbarium records. And there's the web link there where you can see the 2 progress reports for that project. So that kind of just got me introduced to the power of citizen science and how it can really get people engaged. And so I developed a citizen science project on iNaturalist called the Whitebark Pine Project. And so that's linked there. You can join that as a member and you can make observations and they'll show up in that project. So that's just part of the broader Whitebark Pine Project, which is really just a personal project for me. So what are the significance and threats to five-needle pines? So that was covered in that video, but I'll go through it again here and just to mention that they're keystone species oftentimes, meaning that there are other species in an ecosystem depend on it to such a degree that if they were removed, the ecosystem would change drastically. There've been it's been shown that there's increased mortality rates in whitebark pine and limber pine, Pinus flexilis, and in recent years, and western white pine also has shown declines due to pests, fire suppression, and logging. And I just would caveat that the logging piece just to say that not all logging is bad. There's certainly large scale industrial clear cutting is bad, but silvicultural treatments can actually, if they're done correctly, can actually help forests become more resilient to wildfires and to pests and pathogens. For instance, thinning western white pine or limbing up western white pine can help reduce the risk of white pine blister rust. The threats are really pests and disease, climate change, and wildfires are the biggest threats to these species. And on the right is a photo of white pine blister rust. It's caused by a fungus that is not native to North America. It's from Asia. It was introduced to North America via Europe in the early 1900s. So there is an urgent need for data on five-needle pine populations, and there are federal and state programs. For instance, the U.S. Forest Service's Forest Inventory and Monitoring program has been going on for decades. They have a lot of data, and they go back and they repeat the data collection, and so that's how they're able to track trends. And it's certainly very important sources of data. But I also feel there's a role for crowdsourced data from community science, and it is emerging as an important supplemental source of information for conservation and biodiversity research. And that's been shown in several papers in the last 5 or 6 years have come out. illustrating the uses of citizen science or community science data. So what is citizen science or community science data? Well, it's public participation, often voluntary, in scientific research by both scientists and non-scientists. The advantages of citizen science data are that it's freely available. There's established platforms for data storage. For instance, iNaturalist is one example. It's crowdsourced. It's reliant on community involvement, and so you can really scale up. And if you've ever participated in a BioBlitz, you know just how powerful it can be to collect a lot of data fast. And then it increases the public appreciation and advocacy for scientific topics. On the right is a screenshot of another iNaturalist project that I'm involved in. It's the 5 Needle Pines along the Pacific Crest Trail. Of course, when I was in Winthrop, the Pacific Crest Trail goes right near Winthrop in the North Cascades, and so my Whitebark Pine project sort of overlaps with that area. But the folks with the Pacific Crest Trail Association and the California Native Plant Society and a fellow named Michael Kaufman were interested in recruiting essentially the through hikers and other hikers to document 5 Needle Pines along the Pacific Crest Trail. So that's another project that you can check out on iNaturalist. So we talked about the advantages of citizen science. What are some of the disadvantages? Well, unknown or variable quality of observations. So kind of, you don't know what you're going to get. You get a lot of data. Some of it's good, some of it's not so good. There are methods in place, on iNaturalist where you can have people verify your identifications, but you may not get good location data. The data is often unstructured. It lacks a rigorous study design. So as a scientist, I'm always thinking about study designs, statistical power, and if your data is unstructured, you can lack the power that you need to actually say something about the topic of interest. Spatial bias, usually towards easily accessible areas, roads and trails, is another disadvantage. And also, it's that that lacking that study design. So in essence, the lack of quality and rigor in community science data reduces its usefulness for certain applications for scientific researchers. And on the right is a whitebark pine cone, just beautiful, deep purple, just lovely. So I got to thinking over the years here of working on these citizen science projects, and thinking, well, what if we could leverage the power of citizen science while minimizing the downsides? And if we could, how would we do that? So to undo that, I employed a 3-pronged approach of outreach, education, and engagement as part of this pilot project that was funded by Washington Native Plant Society. So outreach and education was through the Whitebark Pine project website. And if you get on there, you'll see there are information about 5 Needle Pine anatomy. There's an identification guide to 5 Needle Pines. There's descriptions of the 9 5 Needle Pines that occur in the Western United States. Lots of different tools out there to just get people to understand more about these important and majestic species. And then there's the engagement piece was this 5 Needle Pine citizen science monitoring project. And on the right is the cover of the report that I produced and provided to Washington Native Plant Society as part of the grant funding. That's available on my website. So the objectives of the project, 4 of those, so to perform local community outreach and education and recruitment of volunteers, to support the data collection on 5 Needle Pines in the North Cascades. Secondly, to prepare a statistically robust study design. So I used the stratified random sample, and that was, to add some rigor to this study. Develop and test field methods for collecting, monitoring data on 5 Needle Pine populations that dovetail with the U.S. Forest Service forest inventory and analysis methods. So my interest there was how could we could we collect data? If we collect this data, it would be great if it could be used to actually supplement data that's already being collected. And so we're collecting the same data attributes using the same domain lists. And then of course, to collect, analyze, and report on the data. Okay, so what are five needle pines? I talked about them in the video a little bit, but we'll go through it again here. So five needle pines are conifer trees in the genus Pinus with needles that occur in bundles. They call them fascicles, and each bundle has 5 needles. So you can see in my hand there, I've got a Western white pine fascicle, and you can see the 5 needles clearly attached at the base. There are 9 species of 5 needle pines in Western North America. What are those? They're listed there on the right. Whitebark pine, Pinus albicaulis, Rocky Mountain bristlecone pine, Pinus aristata, foxtail pine, Pinus balfouriana, limber pine, Pinus flexilis, sugar pine, Pinus lambertiana, Great Basin bristlecone pine, Pinus longiava, Western white pine, Pinus monticola, Southwestern white pine, Pinus strobiformis, and Torrey pine, Pinus torreyana. And you can see the generalized ranges. Just I use states just because it's familiar to people. And then are they a high 5 pine? What does that mean? Well, of the 9 species, 6 are considered what we call high 5 pines. That is a 5 needle pine five needle pines that occur at high elevation. So their habitat is really up above about 5,000 feet above sea level. And as I mentioned earlier, there's a identification guide to 5 needle pines on my website, alphawoodecology.org. So in Washington State, though, what are the 5 needle pines in Washington State? There are 2 of them that are known to occur. If anybody else finds another species, let me know, because I would love it. But pretty well established that there are 2. Western white pine, Pinus monticola. Its habitat is lowland to upper montane and lower subalpine areas. The species often occurs on sheltered slope positions and valley bottoms, and its elevation ranges from 0 to 3,000 meters, depending on where you are, what your latitude is, essentially. And then the range is across most of the Western United States and Canada. Western white pine, similar species would be sugar pine. The primary difference between the 2 being the length of mature seed cones, 15 to 25 centimeters or 6 to 10 inches for Western white pine, and greater than 25 centimeters for sugar pine. So sugar pine's got huge cones. If anybody's ever seen them in California, they're just massive. You would not want them to fall on your head when they're full of sap. Whereas, the Western white pine are smaller. Special status, not really, there isn't much in terms of special status. In Canada, they're not listed under the Species at Risk Act. They're not threatened or listed in the United States. A couple states have them listed as in their kind of their state listing as imperiled, critically imperiled, or vulnerable. It's Alberta, Montana, and Nevada. So not really listed. And here's some photos of Western white pine. You can see the long cones, long narrow cones. So if you're in Washington, though, you've only got really the 2 to think about: Western white pine and whitebark pine. Whitebark pine is the second one in Washington State, Pinus albicaulis. Its habitat is upper montane and subalpine areas up to tree line. Actually, the North Cascades, I've seen stunted, small stunted whitebark pine at 8,000, 9,000 feet up on ridge lines. And across its full geographic range, the species occurs at elevations ranging from 4,300 feet to 12,000 feet. And of course, again, as you get further south, it gets higher. Its range gets higher and higher in elevation. And then it occurs across much of the Western United States and Canada as well. Most similar species is limber pine, where the 2 species overlap. They can co-occur on the same site and can be challenging, especially for the younger saplings where you don't have cones and seedlings. Special status across the board. In Canada, it's endangered. The International Union for Conservation of Nature, the IUCN, considers it endangered on their red list. United States, it's threatened. And then across the states and provinces that consider it S1, S2, or S3. There's a number of them, basically all of the states that it and provinces that it occurs in. And here's some photos. What whitebark pine is interesting in that, you know, cones are deep purple when they're younger, the seed cones are at least. Then when they get older, the as you saw in that video, the Clark's nutcrackers will pull them apart, pull out, and basically what you end up with underneath the tree oftentimes is not much more than cone scales and maybe a couple dilapidated cones where you don't see too much. Whereas if you were in a state where there's lumber pine, you would see intact cones underneath the mature trees. And then at, you know, near tree line, they could be pretty small trees, and but lower down in elevation, they become more of a full-size tree, or can become more full-size tree. And then here's more of that video, because it's just fun to watch the Clark's nutcrackers go to town on the whitebark pine cones. So here's just a fun little video we put together in the North Cascades. So whitebark pine, you know, and the Clark's nutcrackers go hand in hand. They have a mutualistic relationship. The nutcrackers, you know, as you can see, they're really intent on getting at those seeds, and they'll cache the seeds on windswept slopes and ridges to come back for later. I'm going to save a snack for later, right? But they'll forget about some of them, and inevitably some of those sprout into seedlings. And so it's a really neat plant-animal relationship there. All right, so just to jump into the methods, how did I approach this project? Well, I made a call for citizen scientists in the Methow Valley, posted in the local paper, call for volunteers. I looked into used social media, reached out to some folks with the Okanogan Chapter of the Washington Native Plant Society to put out an announcement, and then reached out to friends. And it was about 50-50 friends and friends of friends, and also just some folks in the community who were just interested, who showed up. I had a project ambassador, was what I called that person, who was paid for their time to assist. And this is part of my model I'll talk about a little bit later, how this could be scaled up. Yeah, we reached out to the local U.S. Forest Service Ranger District. Wanted to make sure because we were working on the Okanogan Wenatchee National Forest. That was the idea. We wanted to make sure they were aware and okay with it, and they were. We also reached out to the national, as I mentioned earlier in the acknowledgements, the National Citizen Science Coordinator for the Forest Service and the FIA National Program Manager, to get them on board and check out and review the methods. And then, so what I did was I followed a reduced version of the forest inventory and analysis methods from the Forest Service. And I chose a subset of the data attributes that I've thought that citizen scientists could reasonably record with minimal technical experience and training, and do that in a reasonable amount of time. You know, if anybody's ever delved into the FIA methods, it's extensive. And you can see on the right, there's 4 subplots that can constitute one plot, you know, and there's the outer macro plot and micro plot. And so, you know, thinking about trying to do that with citizen scientists in a reasonable amount of time just seemed like too much. So I scaled it back to one subplot, just the subplot 1 in the middle there. We had the micro plot as well. We sampled seedlings and saplings, or counted seedlings and saplings in the micro plot. And then we just, I selected you know, data attributes, for instance, you know, tree height, tree diameter, breast height, dominant plant species, and so that it could go pretty quickly and hold people's interest. And then we also, FIA methods, they count every tree. They're counting and measuring every species of tree and recording every single plant species that's there. And so we kept it quick with dominant plants, and we also just focused on 5-needle pines to keep it relevant. Keep saying we, but it's the royal we. So here's our, oh, hold on. One sec. Okay. So here's my study area map. You can see down in the lower right corner, there's the kind of the broader, bigger picture. You can see British Columbia and Washington. You can see Winthrop and Twisp. If anybody is familiar with North Central Washington, you'll know about where this is. The red box is where we're zoomed into in the broader map, or the kind of the zoomed in map. And so the green area is actually what my study area was. It's about it's a 10th acre, or no, sorry, 10th mile off of the trail that goes up to Cutthroat Pass, and you can see the Pacific Crest Trail up there near the ridge. And the dots are the sample points. So those were the design points. So those were kind of where we had intended to go based off the stratified random sample design. The different colors of green are the different vegetation communities. So I prepared a vegetation map. And that's what I used for the stratification along with elevations. I had 3 different elevation strata: a lower strata, a middle strata, and an upper strata. And so the idea was to go to those points, and I developed, you know, more sample points than I thought we could probably get to in the amount of time we had, just because some of them were going to inevitably end up on cliffs and unsafe areas, and we'd need to skip those. And you can see the red crosses are where we actually got to. Those are the plots that we sampled. So we went out in July 1 and 2 and July 23 in 2023. We recorded data on smartphones using a custom form in the Fulcrum app. There were 7 people on the team. So the team lead was me, the project ambassador, and then the citizen scientists. 3 field teams. There were 3 field teams of 2 to 3 people each. I tried to, you know, there were varying amounts of, you know, knowledge about plants on the team. So I tried to split up the teams in such a way that there was at least somebody who knew something about plants. And we listed the dominant plant species in abundance, various tree measurements, as I mentioned earlier, and environmental data like slope gradient. And we took photos of the landscape and of various plants. And we utilized the iNaturalist Seek app, so people who didn't know plants very well could take photos and use that to help with identifications, which were later uploaded to iNaturalist, and then I went through and verified those later on and then updated the data. So this is a way to use iNaturalist to kind of enhance your team and help people, you know, learn more about plants. Okay, results and discussion. Well, I'm just going to caveat this by saying that, you know, it was a small sample size, and so, you know, these results really should be put in the kind of that context, in that we shouldn't, you know, may or may not be seeing real trends here. We'll just say that from a scientific standpoint. But the idea is this is a pilot study, and so that ideally this could get scaled up in the future. We'd have a large enough sample size to actually be able to say something. Certainly the study design itself with the stratified random sample design and the standardized approach to data collection would lend to that in the future with a broader study across time. So for 5 needle pines, we sampled 17 plots. We both of the 2 species that are present in Washington were in the study area. There were 56 observations of 5 needle pines, 26 5 needle pine trees. So these are the tree size greater than 5 inches in diameter. 2 of those were dead. There were 4 saplings, 1 to 5 inches in diameter, and 26 seedlings. 5 whitebark pines were damaged from white pine blister rust. You can see on the right there is a photo of the blister rust. And then an unknown foliage disease was commonly observed in western white pine throughout the study area. So that year, I don't know if this happened, wasn't there the year before in 2022. I don't know if it was there in 2024, but nearly all of the western white pine, the needles were orange across the entire tree. So I don't know what ended up happening with that. But that was recorded in the data. And then this map shows, this is similar to the other map, except it's showing the size of the circles are the number of 5 needle pines observed at the plots. The color of the dots in the middle are the species that were present at the plots. And then I also pulled in some observations from other data sets like iNaturalist and herbarium records. So those are the triangles. So you can see where other people had observed 5 needle pines in the study area. And then the color of the circles is the average height of the 5 needle pines in those plots. So the darker the color, the taller the trees. So in general, what we noted was that as you moved up in elevation, the trees got shorter, which makes a lot of sense. Okay, so 3 of the 17 plots had no 5 needle pines present. These were Pacific silver fir communities. So that they're generally cooler and shadier, and both of the 5 needle pines, western white pine and whitebark pine, they like more the early successional species. They're shade intolerant, and so they like open canopies. But yeah, 5 needle pines were most commonly associated with communities dominated by subalpine fir, Abies lasiocarpus, and subalpine larch, Lyrax lyalii, and less frequently observed in associations dominated by or co-dominated by Pacific silver fir. And when I say association, I'm referring to plant associations or plant communities. The distribution of each species in the study area was also consistent with the known life history traits of these species. As I described them earlier in this talk, the western white pine is more in the lower elevations, and the whitebark pine is in the upper elevations and exposed slope positions. So that was consistent with those life history traits. And then the graph on the right just shows the number of so the 2 panels, the left side is whitebark pine, the right side is western white pine, and it's just showing the count of the trees that we observed out there by size class. So tree is green, sapling is orange, and blue is seedlings. And then at the bottom there on the x-axis are the different communities. So you could see, which communities we found the most of the whitebark pines in was the subalpine fir huckleberry community. So for whitebark pine, just jump into specific results for each species. We found 20 live and 2 dead whitebark pine trees, 4 saplings and 23 seedlings. There were a low number of saplings observed relative to tree size whitebark pine, and that's illustrated in the graphic on the right there. We can see the number of live whitebark pines and the seedlings and the saplings and then the trees. You see the saplings number is very low. What this is we're typically what you'd expect would be that you would have what's called a J shape, where you would have more saplings essentially. and typically more saplings than trees. And the size class distribution was not a typical J shape, or sorry, reverse J shape distribution. And that's typical of naturally regenerating forests due to and this is specifically because of those low number of saplings. So I don't know what's going on. It's maybe it's related to the sample size that we had. It's just, like as I mentioned, it's a small sample size. Maybe there's something going on with the saplings there where they're dying off, possibly. Hard to say at this point. And so I mentioned earlier just height of these trees by elevation class. And so you can see, well, first of all, that Western white pine was really only found in the lowest elevation. It was, so that's on the left side is the tree diameter at breast height. And so that's the sort of, and then it's split off by elevation class. And on the right side is tree height. And so the green boxes are whitebark pine and the red boxes are Western white pine. So we only see the red boxes in the lowest elevation strata. And then we can see that in general, the tree height was varied by elevation. For Western white pine, there were 4 live Western white pine trees. We counted no saplings and only 3 seedlings. The Western white pines were observed only in the lowest elevation stratum, as I mentioned, and Western white pines were generally taller and had a greater diameter than whitebark pine in the same elevation strata. All right, so just to talk more about the citizen science monitoring, this study, it demonstrated how citizen science could be leveraged to contribute valuable data using the 3-tiered approach of outreach, education, and engagement. I fostered environmental stewardship in connection with nature. I actually had one of the participants mention, like, I've hiked this trail so many times and I've never looked at the landscape like we looked at it. And it gave her a new perspective, of looking at the plants and identifying the plants and recognizing the different trees. So I felt like that was a big win. We were able to utilize digital tools. We used the Fulcrum software for efficient data collection and integration, which I'd highly recommend, especially, if this was a scale up, especially that would be important. Entering data from paper field forms is really kind of ancient history at this point, and it takes a lot of time. So being able to utilize those digital tools is really key. And then we also used iNaturalist Seek, and then I didn't mention this, it's in the report, but we also recorded wildlife observations, and there's an app called Merlin that you can use to identify birds by their calls. And so we use that app as well to identify birds that we heard. And we also demonstrated the cost effectiveness of citizen science monitoring 5-needle pines. For instance, the budget for this pilot project was approximately $1,500. That comes out to about $100, less than $100 per plot, which is much less than traditional inventory and monitoring studies. And that's not to say that if we scale this up, it might not cost a little more than that, but I think in general, you think you get the picture that it'd be a lot less. So what were some of the challenges we encountered? The challenges, there was a challenge of recruiting project team members. And so, as I mentioned, about half the folks were friends or friends of friends, and just trying to get, in general interest from the community. I could have done better, I think, with maybe posting more, flyers or, getting out there more and going to local schools and reaching out to folks. But we did find our volunteers, and I'm very grateful for them. There's a necessity to balance, that detailed field protocols with volunteer engagement, and from what I gathered from feedback from the participants, I feel like it struck a pretty good balance there. But we did have folks, one person, mentioned that their hand hurt from entering the data the whole time on their cell phone, and got me thinking that I should have sort of advised people to switch up roles more. So that everybody could have a chance to just look at the plants and not always be entering the data. So that was something to think about. It's something I do professionally as a botanist is we switch up who's collecting the data, recording the data, and then difficulty in collecting sufficient data for statistical significant analysis. and power analyses is recommended for optimal sample sizes in the future. Again, this was a pilot study, we had limited time and budget, you know, in the future to scale up, you know, you'd really want to focus on getting that minimum amount that you need. That would be a power analysis would actually tell you that. Scaling up citizen science monitoring. This is, you know, sort of this pilot study. So, you know, in the back of my mind, I'm thinking, how could we make this bigger? How could we scale this up and actually take it to more of a state or regional or range-wide scale? And on the right is the range map of whitebark pine in the western U.S. and Canada. Pretty big area. And so, you know, you'd want to start small, probably, you know, county or state or something, and before you started thinking about it at the range-wide scale. But, you know, you got to shoot for something, right? The advantages of scaling up would be an increased outreach and sample size. Cost effectiveness, again, you know, is a factor. Standardized methods. So if everyone was using the same methods across this whole area, then the data, you'd have even more power in the data. It would augment the FIA data. So then the Forest Service, they could take it and take the data they already have and add this data to it. I also feel like it would, you know, could be driven by local initiatives, and that could get local communities, you know, interested and learning more about their natural environment across a broad area. Challenges to scaling up would be, I mean, there's more than this, I'm sure, but top of mind is coordination and funding would be critical. You know, scaling up would necessitate coordinating various local projects, you know, managing more data, maintaining a centralized database, which would lead to increased management time. And scaling up would also necessitate working across jurisdictions, which, you know, could require a coordination with state and federal agencies across those jurisdictions. How could you scale? What's some ideas for how you could approach this? You know, you'd want a project manager probably, or a couple of project managers. This would be a dedicated, you know, team to, you know, increase the oversight. Although, you know, one project scientist and manager could cover large areas, because you're actually relying on the citizen scientists themselves and your local ambassadors to do a lot of the heavy lifting on this. So you would need, you know, fewer folks higher up in the hierarchy there, if you will. You could, you know, take a bottom-up approach, empowering local ambassadors to build teams, and which would simplify the management. So you'd be, you know, coordinating with the ambassadors who themselves are on the ground, you know, in the communities, drumming up interest and getting out there on the ground. And then you get 1 to several project scientists, 1 project manager, local ambassadors and volunteers. The local ambassadors would be paid positions potentially to keep them engaged in actually, you know, working and be a seasonal or part-time position. You can engage primary and secondary schools, colleges, and universities to encourage student participation, possibly graduate students at the collegiate level. And then you could manage your data digitally using a centralized database. So if anyone's familiar with the Fulcrum app, you know, it's a way to collect data on your smartphone. The data gets uploaded to a database on the cloud, and it's all in one place. So no matter if you're in Nevada, you're in Washington, wherever you are, the data is all going to the same place using the same app. So that's some ideas. One application idea I had was for the Whitebark Pine Ecosystem Foundation. It's a great organization. If you don't know about it, you should check it out and consider joining or donating. But they have this Whitebark Pine ski area program certification, and it's you know, basically recognizes leadership in whitebark pine initiatives and raises awareness of the challenges of sort of that the 5 needle pines face, specifically whitebark pine, and engages the public. And it's something that any ski area can apply for, and there's criteria. So one of those criteria is that there are some kind of monitoring and mapping of whitebark pine. And so this approach that I've outlined here, this pilot study, could maybe be, you know, one application of this, or this whitebark pine ski area could be one application of the approach I lined out. So in conclusion, citizen science initiatives for 5-needle pine monitoring is a cost-effective approach to enhancing awareness and understanding of these trees in local ecosystems, while also providing essential data for their populations. The local scale efforts can act as an early warning system for identifying threats to 5-needle pines. For instance, when we observe the orange needles on western white pine, that's maybe something that the Forest Service didn't realize right away. Just as an example. And then at broader regional and interregional levels, citizen science can significantly raise awareness of the conservation challenges facing 5-needle pines and complement official monitoring data sets. So there's my contact information and my website again. And I will open it up for questions.
Wow, thank you, Aaron. That was great for a small scale project. There's certainly a lot of information and food for thought there. That was really great, and I love the beautiful pictures of the Cutthroat Pass area and the whitebark pine. We do have a couple of questions. One of them, I think you already answered with your presentation, was how many were western white and how many were white bark, and you showed a graph of that, but perhaps yeah, you could show that again. Okay.
Yep. So the whitebark pine, what is that? 47 whitebark pine across all of the different height strata, and then 4 western white pine trees and 3 seedlings, so 7 western white pine. So mostly we found whitebark pine. I think largely because the study area itself was started pretty high in elevation, but the trailhead's pretty high up for western white pine.
I was also wondering about the diameter, I guess, with a tree being more than 5 inches diameter, but so many of them are multi-stemmed up high. And that would make it hard to quantitate a total diameter, I would think.
Yeah, I mean, each stem is measured separately when you have multiple stems. There's a height threshold when you have multiple. If the multiple stems start at a certain height, then you count them as a separate stem. Essentially, that's how the FIA methods do it. Yeah.
Okay, good. There is a question about the western white pine. What birds or insects depend on the western white pine? We talked about Clark's nutcracker and the white bark, but can you speak to the western white pine?
No, I don't think I can speak to that very well, to the species. I know that I know for certain squirrels will eat the seeds. If you've seen the middens that are below, western white pines, but for birds, I don't know, actually. Yeah.
That's a good thing to find out. Great.
Yeah,
Great.
It sparked my interest.
Good. Okay. Let's see. Oh, good question. Will you be recruiting more volunteers this summer? And she also asked if you will be doing any hypothesis testing as well as a descriptive study.
Yeah, I don't have plans this summer to continue this study. The funds have, been used from the grant. And so, I'm currently, I guess I'd say my status is looking for interested folks who may be wanting to fund a broader effort or additional years of effort or something like that. And if that was the case, I'd consider, doing that. So yeah, as a professional scientist, it's and a field scientist, it is a busy time of year in the summer for me. So that's but this is a passion of mine. So it's would love to do more of this stuff. So for sure. If I do it, I will reach out through Washington Native Plant Society and we'll get an email out to the members so people are aware that it's happening, if it does happen in the future.
Oh, and here's an interesting question about Yellowstone. In Yellowstone, it is said that bears rely on the seeds from the whitebark pine. Could that be true also in the North Cascades?
Definitely, yeah. There are more than just the Clark's nutcracker rely on the whitebark pine cones, and in places where where bears don't have another ready source of protein, like near the coast or something where there may be salmon or something, that's a huge source of protein for them. So yeah, anywhere there are bears and whitebark pine, they're going to be an important food source.
Right. Right. And another question about the foliar disease that you noticed affecting the trees in the study. Any knowledge of what people might be doing to answer that this summer?
That I don't know. I think the first step would be to see if that reoccurred, if it's reoccurring, if it's still happening, if it's led to some widespread mortality, or if it was just a one-off 2023, thing, maybe a particularly wet winter or something, or I don't know, but I'm not aware of it. I did send the report on to the Forest Service and the Metro Ranger District, and so that they're aware of it.
Well, we can hope that the interest in the resources might be there.
Definitely, I'd say if you're in the area, go for a hike up Cutthroat Lake Trail, and take some photos and post them on iNaturalist of the Western White Pine, so we can all see it. Yeah.
Okay, let me check the chat. Oh, here's a question about in the chat. How resilient are the 5-needle pines once they have the blister rust? Does it shorten the tree's longevity by a lot, or what's the timing on that? Resilience.
Yeah. I'm no expert in that element of it, in the how the disease affects them. I do know that there are some whitebark pine that are resistant, naturally resistant to it, and I know programs out there where people are trying to identify those that are resistant and save the seeds so that they can regenerate them and plant them, so that there are resistant strains out there that I'm aware of. Otherwise, I don't know beyond that.
And there's a question in the chat about early studies of, quote, pole blight, POLE blight, in the early part of the 20th century. Pole blight and blister rust, would those be possibly the same?
I don't know. I can't speak to that. I'm not sure.
Yeah. I think in the Whitebark Pine Foundation website, I think there's more information about blister rust and the seedling regeneration projects. I think that's where I saw some of that about trying to make more resistant genetic strains. I think that's where I saw it.
Absolutely. Yeah. They've got a ton of information on their website, and I'd recommend everybody who's interested go check it out, because you can learn a lot from them. Yep.
Great. Great. Good. Well, I think the Q&A has we've covered it, and oh, here's another comment about organizing for scaling up. Organizing a study forest by forest seems natural. Perhaps some friends of organizations of certain ranger districts that could be useful, but again, it does rely on funding. But yeah, those are some ideas for spread and scale.
Oh, for sure. Yeah. No, it seemed to me like a natural way to go would be like identify where are all the Forest Service ranger districts on forests that have whitebark pine or 5-needle pines in general, and reach out to them, start with them as a resource. And then I would also say that, I spoke a lot about funding, but there is I don't want to discourage anyone who wants to go out and do this on their own. This is, isn't something that, it's basically something that anybody could do. Like if you're interested and you go out and you learn about these species, and it could be just as simple as taking photos of these 5-needle pines and posting them on iNaturalist so we know where they are. Just as simple as that. It doesn't have to be as elaborate as what I've shown here, or it could be as elaborate or more. But I think you all on this call and everyone has the power to, if they're interested, to go out and do that and do something meaningful to help these trees. So please do it. Please get out there and get your cell phones going and get an iNaturalist account if you don't have one, and yeah, start posting those photos.
Great, great. Well, thank you again, and that's a great way to summarize and engage everyone in learning more and possibly helping get some more information. And knowledge is power, so that's a great way to go. I would like to just end by thanking you, Aaron, very, very much. And I would also like to thank everyone who's been on this call. And again, if you check the Native Plant Society website, click into the bio blitzes, which are occurring this week in pretty much all parts of the state. You might find one nearby. Here, Denise is showing us the screen. That's great. You can also just, again, get onto iNaturalist and start learning how it works, because it is a great tool. And this is a great picture of the observations that have already been done. The Central Washington chapter is going great guns. And our little project on Saturday up in the Columbia Breaks Fire Interpretive Center along the Columbia, we're going to be learning together how to post our observations and doing more checking on the identifications and learning how to do it. So it should be a really fun end to Native Plant Appreciation Month, and give us all tools we can use for the future, for future Native Plant Appreciation events in the future, I hope. So, Denise, did you want to add anything to that from the statewide perspective?
I just want to thank you all for being here tonight, and happy Earth Day. Get out there and try iNaturalist. And Aaron, I just want to thank you for your project. Thinking about the whole concept of organizing has made me reflect on putting our BioBlitz together and getting folks involved. And it is a process, but oh my gosh, what great rewards we can get from the experience. So I wish everyone a wonderful time that's out there learning for the first time. And thank you to those experts that are posting like crazy. Do log in, check out our project, look at everyone's beautiful photos, and enjoy. In the May e-news, we'll send out some results just from our experiment of how many folks can we get involved in this BioBlitz project. So thanks for being here. Very good.
Thanks, everyone. Bye-bye.
Thank you, Aaron. It's a pleasure having you and hearing about your project. Good night, everyone.
Thanks. Good night.